Welcome to the video for chapter 5.5. This is our last video that's going to be covering bacteria slash prokaryotic cells. In 5.6, we're going to do a quick summary about some eukaryotic cell characteristics, but chapter 5.5 is a really fun one. We're going to talk about specialized structures in bacteria, so structures we don't find anywhere else. Okay, but before we start into 5.5, let's do a review question. So, how is DNA most commonly arranged inside a bacterial cell? A, multiple pieces in the cytoplasm. B, a circular molecule tangled throughout the cytoplasm. C, a circular molecule with loops bound by DNA binding proteins. Or D, a circular molecule with a nuclear membrane. Okay, so pause the video and see if you can find the answer. Okay, so let's go through this question. The answer is C. So let's go through this piece by piece and from what we know about bacterial cells and their DNA. Well, there's one of the answers here that we can immediately get rid of. Okay, and that's the circular molecule with a nuclear membrane. Okay, bacteria are prokaryotic. They don't have membrane-bound organelles. They don't have a nucleus. Okay, that's what nuclear membrane is hinting at. Okay, so that's one down. Another one we can immediately get rid of is A, multiple pieces in the cytoplasm. The cytoplasm part is correct. That's where bacterial DNA is found. It's found in the nucleoid region in the cytoplasm. The key here is multiple pieces. Okay, we know bacterial cells have one single circular DNA chromosome. Okay, so it can't be multiple pieces. So that eliminates A and that eliminates D. So that leaves us with B and C. Now, we talked about in class how the single chromosome for bacteria is going to loop on each other, and these DNA binding proteins are going to keep it looped so it can stay concentrated in that nucleoid region. Okay, so C is the correct answer. B, what this is meaning is that it's just allowed to kind of free float through the cytoplasm. And we know that it doesn't do that. We know that the DNA, as much as possible, clumps together in groups in that nucleoid region. Okay, let's talk about the section objectives for 5.5. You need to be able to describe how pili and stalks enable bacteria to adhere or stick to a substrate where conditions are favorable. And you need to be able to explain how flagellar, motility, and chemotaxis enable bacteria to respond to environmental change. There's so many fun things in this chapter, guys. I'm excited. Okay, so we've talked about this before. Bacterial cells are going to respond to their environment. In fact, bacteria cells, if they sense with those receptors in their membrane that the environment isn't a good one, it can do several things in order to survive. But the biggest one is it's going to leave that bad environment. But we really don't talk about what happens when they find a good environment, what we call favorable conditions. Okay, so when bacterial cells find an environment that has lots of nutrients, it's going to make them be successful bacteria. They're going to grow and divide and multiply. Well, bacterial cells want to stay in that type of environment. They don't want to leave it. So what they're going to do is they're going to adhere or attach to specific surfaces in that environment and just flourish. Okay, so we define adherence as the ability to attach to a substrate, aka we mean it as a surface and not a solute in this case, like rocks in a nutrient-rich pond or a cell in a nutrient-rich host. Okay, so it's the ability to attach. Okay, when these bacterial cells adhere, they increase their chance of survival. Okay, so, and as we said, in other environments, like if it's a bad environment, so an environment can start out as good, right? Just like it can for humans, but a environmental change or a natural disaster can change the conditions so that they're inhospitable for bacteria as well as humans. Okay, so when the environment changes and the cells can no longer survive there, they need to leave. They need to unadhere from their substrate and they need to literally relocate. So we call this ability to move and relocate motility. Okay, so a large part of this chapter section is going to be about motility structures, so the structures the cell uses to move and how it uses them to move, and the structures that they, that bacteria use to stick to surfaces. Okay, so the first specialized structures we're really going to talk about are the pili and the stalks. Now, pili should be a little bit familiar. We encountered them in Biology 201. We talked about them. Pili have a couple different functions. Their main function is to help the bacterial cell attach to a surface, okay, help it adhere. Okay, pili is the plural form of the word. Singular would be pilus. Um, Pili are made of pilin, so that's the protein that makes them up. Pilin's so original. Scientists are really great at naming things. Okay, we have different types of pili, and we'll actually talk more about different pili in a different the different types of pili that there can be in a different lecture. But let's talk about some of the general forms of pili. Okay, so we have fimbri. Fimbri are pili, but they're short. Okay, and fimbri only have one job, and that is to literally help the cell stick to different surfaces. Okay, we have a special form of pili, which are called conjugation pili. Sometimes this is referred to as a sex pilus. That's kind of a misnomer. It's kind of misleading because bacterial cells don't participate in sexual reproduction. It's not really a sex pilus. Okay, so this conjugation pilus is going to be really important. This conjugation pilus is a way that a bacterial cell can swap bits of DNA with another bacterial cell. Okay, so if we think about this, say you have a cat and you take the gene from the cat for its cat ears and swap it back and forth. That's kind of what happens between bacteria with this conjugation pilus. Instead of getting your cat's ears, what's going to happen is they're going to transfer things called plasmids, which are tiny circular pieces of DNA that aren't part of the actual genome. And these plasmids are going to be for genes or a gene that are going to make specific proteins. Now, we actually are really concerned with plasmids because we think this is how microbes or bacteria specifically have become antibiotic resistant. And the more they exchange these plasmids for antibiotic resistance, the more and more bacterial species we won't be able to treat with our current antibiotics. Okay, so this conjugation pilus is a very, very important pili. But as I said, we'll come back and talk about the different types of pili in a different lecture. We're just going to talk about what's here in general. Okay, so the last thing we're going to talk about is the stalks. So pili are made of the pilin protein. Stalks are actually extensions of the cytoplasm. Okay, so it's going to kind of make a sticky substance outside of the cell. It's going to allow the cell to stick to different surfaces. So the stalks themselves are going to excrete, so they're going to give off adhesion factors. So they're going to make these chemicals that are going to help even more the cell stick to a surface. Okay, so if we look here at this micrograph, you can see these white lines outside of the cell. Okay, these are the stalks. Okay, this is those cytoplasm extensions that are going to facilitate the bacterial cell sticking to a surface. Okay, so we have this cartoon structure. It's going to illustrate some of our different motility structures, the ones that I really want you to know. So the pili are really important. The fimbri are really important, but also is the flagella. So if we look here at our little cartoon diagram, we have these long purple guys. This is the pili. Pili is always longer than the fimbri. Fimbri are short little guys. Remember, they just help the cell adhere to things. But another motility structure that we talked about back in Biology 201, along with pili, is the flagellum. Okay, this is the single form of flagella. Flagella is the plural. Okay, this flagellum is what is the main motility structure for bacteria. Okay, so both eukaryotic cells have flagella. So an example of a human cell that has flagella is a sperm cell. Okay, both eukaryotic cells have flagella, and so do prokaryotic cells. Now, there's a difference in the flagella between the two. So, bacterial flagella are rotary. Okay, so that means that they're going to kind of spin like a propeller. Okay, eukaryotic flagella are going to have what is called a beat pattern. So they're going to go back and forth instead of turn like the bacterial flagella. Okay, in fact, bacterial flagella is rotary. They have this structure that kind of looks like a rotary motor that allows the flagella to turn. Now, this can turn both clockwise and counterclockwise. It's going to impact which direction the cell goes. But we have this um, electron micrograph here, and it really does look like a little motor, like we put a little propeller onto a bacterial cell, which is really cool. You don't need to know the different parts, but you do need to know that bacteria flagella are rotary, so they turn in like a propeller. Okay, another thing that we need to talk about is a lot of bacterial species are going to actually have multiple flagella that are going to work together to move the cell. Okay, these rotary flagella, whether it be a single flagellum or multiple flagella, have one goal, and that is to enable motility in something we call chemotaxis. Now we're going to talk about chemotaxis here on the next slide. Okay, so chemotaxis, chemo means chemical, taxis means movement. Okay, so chemotaxis is literally when a bacterial cell is going to use those receptors on the outside of its membrane to detect different chemical signals in its environment so it can find those good and bad environments. Okay, so it's going to use chemotaxis to move towards nutrients, and it's going to use chemotaxis to move away from things like toxins and antibiotics. So let's look here at these pictures. These are really cool. Okay, so we have our little bacterial propellers. They're so cool. So most of the time, a bacteria cell, the flagella are going to be going in a counterclockwise. So whenever in a science book you see CCW, it just stands for counterclockwise. Okay, so it's going to be moving in a counterclockwise. So it's going to be turning this. Oh, I'm backwards with my computer. So it's going to be turning this way. Okay. And that's going to allow it to move forward. Now, when a bacterial cell wants to change directions, it has to change the direction the flagella are moving. Okay, so it's going to kind of stop, and it's going to start rotating its flagella in the opposite direction. So it's going to be going in the clockwise direction. Okay, so when it starts moving in the clockwise direction, it's going to cause the bacterial cell to do what we call a tumble. Okay, so the cell's going to tumble and it's going to change direction. Now, bacterial cells move by semi-random movement. Okay, remember, bacteria, while they are highly evolved, they're not very smart, right? They don't have a brain, they don't have a nervous system. So all they can do is use these receptors on the outside of their cell to move towards good environments and away from bad. Okay, so we're going to have this little bacterial cell. It's going to have its flagella moving in the counterclockwise direction. So it's going to start swimming towards a attractant, but maybe all of a sudden it detects chemicals in its environment that are bad. Okay, so we're not in a good place. We need to change direction. So it's going to start to move its flagella in the clockwise direction. This is going to actually cause it to turn and change direction. So it's going to tumble. It's going to kind of go in this direction. We don't really sense anything this way. So we're going to turn clockwise again. So we're going to go this way. Whoop, we didn't really find anything. We're going to go back up. Still can't find anything. We're going to tumble again. We're starting to detect something, and that's going to swim towards the attractant. Okay, bacterial cells can detect if they're on the wrong path or if they're in the wrong environment. Okay, these bad chemicals in the environment binding to the receptors is going to signal the fact that they need to turn. Okay, and then they're going to switch the direction that their flagella are moving in order to do that turn. Now, Do we know what direction they're going to turn? No, that's the random part. Okay, they're not always moving randomly, which is why we call it semi-random movement. But the direction that they're going to, when they change direction, we don't exactly know what direction they're going to go in until they tumble. Okay, this is really cool. We're actually going to watch a video. We're going to do it in class. I did post it. It's going to be found on the Canvas page. We'll talk about it here in a couple slides, but it's going to show you how bacterial cells move. And it's really, really cool. Okay, so we have different types of chemotaxis. Okay, we have positive chemotaxis and we have negative chemotaxis. So positive chemotaxis means we're going to move towards the good environment. We're going to move towards an attractant, so things that the cell wants to have. And we have negative chemotaxis, which is going to cause us to move away from things like antibiotics and toxins. So we're going to move away from anything that's repellent. Okay, what's really cool is we have some bacteria that can detect magnets. Okay, so we have some bacteria that go through what we call magnetotaxis. Magnetotaxis is literally when a bacteria can orient themselves to a magnetic field and use it to swim in a very particular direction. Okay, in order to do this, bacteria have to have what are called magnetosomes. Okay, so they're going to be little pockets of magnetic crystals that are found inside the cell. So if we look here at our bacteria cell here, we can see these little red guys, these little circles. Those are the magnetosomes. Okay, so they're going to contain the mineral magnetite. Okay, it has iron in it. Iron is naturally magnetic. Okay, so these magnetosomes are going to help this bacterial cell align to the nearest magnetic field. Okay, so to do this, the bacteria are going to swim downwards towards magnetic north. Okay, and this is how they can orient themselves. Okay, this is actually really important. A lot of anaerobes, so bacteria that can exist where there's no oxygen, they're going to live at the bottom of the ponds where oxygen is pretty low. So to help them to get to their anaerobic environment, so an environment without oxygen, they're going to use those magnetosomes to align to magnetic north, and it's going to cause them to swim down to the bottom of the pond. This is going to help them be in an environment where they can survive. Okay, these magnetosomes are really cool. So that video I was talking about is all about magnetotaxis. In fact, it's on Canvas and it's labeled the magnetotaxis video. Now, I can't show it in this slideshow because the lab group that did it out of the Institute of Korea, they posted it on YouTube, and YouTube flags the video if I have another video from YouTube in it. Ask me how I know that. So here is the URL for it, and as I said, I have it posted on the Canvas page underneath this lecture, so you can go and click on it and watch it. We will be watching it in class, but if you miss class, it's a good way to watch it. So they actually get a magnet and they attach it to a video game controller, and they can literally, with that magnet and that game controller, they can literally control in which direction the bacteria move. It's really cool. I will geek out a lot about it in class. Okay, so go and watch that after you get done with this lecture video. Okay, so the last specialized structure we're going to talk about, have talked about quite a bit and referred to it, but some bacterial cells, specifically Bacillus and Clostridium species, are able to make an endospore. Okay, so endospores are protective heat and chemical resistant shells that the bacteria cell can shove its genome in, and when it does this, it can go dormant, and then it can live in a bad environment for a long time, and can reanimate itself when the environment gets good. Okay, so bacteria that make endospores will click this pathway on when the environment gets bad. Okay, so we can do an endospore stain. Now I need to apologize because I keep saying malachite green with the acid fast stain. That is not correct because sometimes I confuse the endospore stain with the acid fast stain. The acid-fast stain uses carbofuchsia, so it gives it that red color. So the endospore stain is going to use malachite green. And we're going to use it as a differential stain. So we're going to add malachite green. It's going to only bind to the endospore. And then we're going to use a decolorizer, and we're going to do a counter stain to get these lovely pink and green bacteria you see here. The green is the endospores. Okay, these endospores are very long lasting. They can survive for a long time. In fact, we literally found bacterial spores in amber that they had dug up out of the middle of the earth. Okay, like million-year-old bacterial spores in amber. And scientists were able to extract them from the amber, and they reanimated them. Like, they brought them back to life. It's literally like bacterial Jurassic Park with the amber and everything. But that's how long endospores can last. Okay, so when they go dormant, they can last in inhospitable conditions for a long time. Okay, so this is what a electron micrograph of the endospore. Okay, we're going to have our core. This is where our DNA is going to go, and this is going to be that outer spore coat that's going to allow it to be both heat and chemical resistant. Meaning, if you boil water with a bacteria that has the capability to make an endospore, it won't actually kill the endospore. Chemicals, bleach, all that can't get rid of them either. In fact, there's only one way to get rid of bacteria that make endospores. Okay, and that is to use an autoclave, so use both pressure and heat to kill them. Okay, we have to fully sterilize the things that they could possibly be on. Okay, bacteria that form endospores are very medically dangerous. We've actually seen this in multiple cases with our case histories that we talked about at the first of the chapter. We talked about Bacillus anthracis and how it makes an endospore. Those endospores can live in the soil for a long time until they get stirred up, say, by a farmer, and then they can relocate to somewhere that has a better environment when they're spun up like that. We talked about the Clostridium botulinum, so the bacteria that was in the can that made those 2 kids in Oklahoma sick. Okay, Clostridium botulinum is going to release the botulism toxin. Okay, and then it just goes all downhill from there. But mainly, the only bacterial species that can form endospores are the Bacillus and Clostridium species. Okay, not many others can. Okay, so here's a diagram of an endospore stain. Okay, so we're going to put our sample on the slide. We are going to use the primary stain, because remember, this is a differential stain. Even though we're not differentiating between 2 different bacteria, we're differentiating between the endospore and the still living part of the bacteria. Okay, so we're going to add the malachite green. It's going to be our primary stain. We're going to apply heat. This is going to be our mordant. Like in the gram stain, iodine's our mordant. What this is going to do is this is going to kind of soften the outside of the endospore coating, and it's going to allow the primary stain to get into that endospore coat. Okay, and then we're going to use water. This is going to be our decolorizer, so it's going to rinse away. So the only thing that's going to be left stained is that endospore coat. Okay, and then we're going to finally use our counter stain, which in this case is safranin. Safranin is going to be applied to it, so the bacterial cell is going to be labeled pink. Okay, so endospore green, cell pink. So we see here in this example that sometimes you can have endospores that aren't still connected to a cell, and that's because they have gone fully dormant. Okay, and then we can have cells that are not making endospores, and then we can have the cells that are currently going through that process of making an endospore and going dormant. Okay, I would definitely know how endospores are medically dangerous, and I would know this endospore stain technique. Okay, so that is chapter 5.5. So we talked about how cells can adhere to different surfaces. We talked about motility. So we talked about how bacterial flagella work. We've talked about chemotaxis. And then we we talked about the bacteria that can form endospores. Okay, so we will see you in the video for chapter 5.6 where we talk about eukaryotic cells.